Well, first I want to thank the organizer. Really, it's a very nice meeting and I'm really happy to be there in real. And since I began to be a young student in a lab, I've been interested in epigenetics. And when I began to do my own research, I was more interested in the link between metabolism, nutrition, and epigenetics. And so this is a slide that I really like to show to my students because that's a nightmare for them. It's the energy metabolism. And as you can see there is two partners in this energy metabolism that are very important. They are called the acetyl-CoA, which is the universal donor of acetyl group to acetylation of histones, and the one-carbon metabolism with the S-adenosine methionine and the folate cycle that are very important for DNA and histone methylation. And so in this paper here, they have done a whole paragraph linking energy metabolism and epigenetics. And as you can see there is a lot of partner of energy metabolism that are linked with epigenetics, one-carbon metabolism, of course, the Krebs cycle giving acetyl-CoA, which can be also obtained by degradation of proteins, beta-oxidation of lipids, and glycolysis and gluconeogenesis from carbohydrates. And all that, and NAD+ also is important, and all that will act on histone and regulates histone demethylation, methylation, or DNA methylation, acetylation histones, but also a huge different other epigenetic marks that are less studied than these three marks here. So in this figure here, we try to imagine what can be the pathway linking the environment and the epigenome. So of course there is this epigenetic machinery, activation or inhibition with the S-adenosine methionine and acetyl-CoA substrate, but also the inhibitors of the HDAC. And there are other possibilities also, like actually the 7 transmembrane receptor linked to protein G are able to have a cascade of signaling where the last partner of this cascade would be an enzyme of the epigenetic machinery such as HDAC that can translocate from the nucleus to the cytoplasm by phosphorylation. And therefore, in this case, you would modify the target genes of that signaling cascade. As regard to nuclear receptor, they do not only activate the transcription machinery, but they also recruit the chromatin modifiers and the remodeling machinery, which make that this modification of the chromatin structure will keep the memory of the signaling cascade from the ligand activated nuclear receptor. And here we also wanted to talk about the mechanotransduction of the signal. The cell is a continuum from the extracellular matrix that is linked to the plasma membrane, that is linked to cytoskeleton, that is linked to nuclear envelope and the chromatin. And you can imagine here that if you modify one of these partners, you will modify this, what we call the mechanotransduction. And I will give you one example. When the mother has an enriched environment and enriched diet in omega-3, this modifies the omega-3 enrichment in the brain cell membrane in the fetuses, and this will last forever in these little rats when they become an adult. And one of the things we are thinking is responsible for that is the fact that this membrane structure modification changes the chromatin organization and modifies the gene expression in the nucleus of the brain of the rat. And particularly, there are an enzyme that is linked to the incorporation of omega-3 in the cell membrane that is upregulated in this brain of the rat. So you can see here that there are very different pathways linking the environment, nutritional environment, and the epigenome. So coming to developmental origin of health and disease, not only the individual genetics and lifestyle can have an effect on its phenotypes, such as a non-communicable disease like metabolic syndrome, but also the environment during development. And we are particularly interested in maternal diet, such as Suzanne Ozan yesterday. And the way this can modify the predisposition to this non-communicable diseases via the nutrient transfer defect via the placenta, leading to organ developmental defect and later on organ dysfunction. But also, this maternal diet can have effect on the epigenetics pattern of the children, and this will lead to the predisposition to the metabolic syndrome. And of course, the lifestyle all around the life will have also an effect on epigenetics, leading to a vicious cycle and the appearance of the phenotype such as metabolic syndrome. So why is epigenetics so important in the DOHAD theory? Because actually there is a major reprogramming of the epigenome during development with this different time window, gametogenesis with the writing of the gametic epigenome, early development with the erasure of this gametic epigenome, and during fetal growth and organogenesis and the postnatal period where there is the maturation of two of the major systems, which is the brain and nervous system and the gut system, digestive system here. During this two-time window, there is the writing of the epigenome in each cell type during cell differentiation for the acquisition of the specific expression profile. So you can imagine here, if something happened in the environment of the father or the mother, this can modify the predisposition to health and disease. And as the epigenetic marks are copied from the mother cells to daughter cells, this can keep the memory to be the support of the memory of the early environmental exposure. But I've been asked a question, how from this early environment to the adulthood, this information will be kept. So I try to figure that out in this cartoon here. So you have, during the development, either an optimal environment, here in green, or a suboptimal early environment, leading to an optimal epigenome or a suboptimal epigenome. The optimal epigenome will make you an adult in good health, while the other make you a predisposition to disease. And the first way would be the faithful copy of the epigenetic pattern, or a secondary epigenetic process that conserves the chromatin structure from the early environment to the adult cells. Another way is that actually the epigenome will modify the way you react to your environment. So there is an iterative process that makes that every time you encounter a specific environment, you will react differently and more, you will be more impacted with a suboptimal environment than an optimal early epigenome, leading to diseases where the optimal environment will give you not so bad phenotype in the adulthood. And finally, there is also interaction with aging, as for example, the team of Suzanne Ozan and Miguel Constancia has shown, that with aging process, there is a relaxing of the chromatin loop that you can also see in a suboptimal early environment, such as low protein diet to the mother during pregnancy. And therefore, this interaction with aging will make that the epigenome of the rats here were more relaxed and were already aged at early stages after birth. So going back to epigenetic nutrition and metabolism, all what I performed as animal model show me that when you check at differences between sex in the offspring, actually you will find sex differences in the offspring. There are sex specificities in DOHAD in mostly all the animal model and human studies you can read. And sex specificities is significant differences between male and female for a parameter. For example, gene expression differences, which is not the same that sexual dimorphism would be a difference in shape. For example, the men having beard that the women don't have. And sex specificities can also be the observation of a phenotype specifically in one sex. An effect would be significant in female, but not in male, and vice versa. And actually, since the year 2010s, there is an increasing interest for these differences between the sexes. I will just come back a little bit on the central role of the placenta in fetal growth. Placenta is really, really important because it's the maternal-fetal interface. It's transfer the nutrients and oxygen from the mother to the fetus and excrete the waste from the fetus to the mother. It's also a very important immune barrier. And here it's a cartoon I found on internet by a French woman that was just telling the story of the delivery of her baby. And she found very strange that the obstetrician was not very interested by the baby, but very, very interested by the placenta. Actually, this is what happens when an obstetrician has read all the paper of David Barker. They know that placenta can be seen as a programming agent because the placenta weight and shape at birth are marker of cardiovascular diseases in adulthood. So in my lab, we are very interested in placental development, and I will present you here the results of my colleague, Anne Couturier-Tarade and Delphine Rousseau. And they have done a rabbit model because actually the rabbit placenta is much more closer in structure to the human placenta than the rodent's one. And they give a high fat and high cholesterol diet from puberty until for 6 weeks, and then during gestation and lactation. But I will stick to the fetuses just before birth. And they check at the placenta. What they saw is that in male, the fetuses are dyslipidemic, while female fetuses do not show dyslipidemia. And in fact, when they checked at the placenta composition in lipids, they observed that in male, there is an increase in triglyceride in the placenta, but in female, what is the most striking is that there is an increased storage and membrane fatty acids. So, which means that here, the female placenta will stock the lipids coming from the mother, while in male, the same lipids will go through the placental barrier and go to the fetuses, leading to the dyslipidemia. So there's a specific placental metabolic adaptations in female that protects the female fetuses from the severe dyslipidemia. In our lab, we wanted, but a rabbit is not very good model for studying epigenetics because the genome is not well defined. And we were more interested in doing experiment in mouse. So we were wondering, is the placenta development affected by maternal nutrition, and are there alterations of the epigenome and epigenetic machinery? So we developed a mouse model where we changed the diets of the mother when we see the plug, which is the beginning of gestation, to either a control diet with 10% lipids and a matching high-fat diet with 60% lipids. And we collected the fetuses and placenta at E15.5, which is the middle of the fetal period. And what we observed is that there is a decreased fetal-placental ratio here, as the pink is the female fetuses, and the blue is male fetuses and placenta, and the light color is for control diet and dark color for high-fat diet. So, in fact, there were no difference in the weight of the fetuses between the 4 groups, while there were differences with the diet effects in the placenta. High-fat diet gives higher placenta, and there is a sex effect with male placenta were bigger than female one, which is quite common in C57 black 6 mouse. And therefore, there is a decrease in placental efficiency, so fetal-placental ratio, either with sex, the male placenta are less efficient than female, and also there is a diet effect with decreased efficiency with high-fat diet. We studied the transcriptome of those placenta, and first we wanted to see whether there were differences between male and females. And we studied here in green, the male and female differences in the placenta, and actually male and female placenta do not have the exact same transcriptome. And there is almost 100 genes in our study that were differentially expressed between male and female placenta. And here in green, it's under control diet, and in yellow, under high-fat diet. So what you can see is the differences between sex are dependent on the maternal diet. And there is only 11 genes here that are differentially expressed between male and female in the two diets, and they are mainly associated to X and Y chromosome. Regarding the high-fat diet effect, we noticed that 164 genes were differentially expressed in female and 187 genes in male. And again, interestingly, the gene sets are not the same. There is only 16 genes that are always differentially expressed in male and female according to maternal high-fat diet. So the transcriptional response to the maternal diet depends on the sex of the fetus. And moreover, if you look at the biological function associated to those genes, they are linked with amino acid metabolism in female and more to vascularization and glucose and lipid metabolism in male. So not only the gene set are not the same, but also the biological function affected by maternal diet are not the same if the fetus is a male or a female. So we wanted to see that is there alteration of transcription and epigenetic machinery in those placenta? And we checked for the enzyme, the epigenetic machinery gene, and we noticed that there is downregulation of the expression of the H3K9 trimethyltransferases that are called KMT1A and 1B. And interestingly, KMT1A is downregulating in both sexes, while KMT1B is only decreased in male. We need not see any global modification by Western blot in this epigenetic mark, but actually Western blot is not a very sensitive technique. We studied also DNMT3L, which is a cofactor of the DNMT3s, and we noticed that there is downregulation of DNMT3L according to maternal diet only in female here. And interestingly, when we looked at the global DNA methylation with a technique called LUMA, we saw that there is a downregulation of the methylation in the female placenta when there is a high-fat diet, which is linked, therefore, to associated to DNMT3L downregulation. Epigenetics is not a static system. It's dynamic and specific process. So to better characterize our model, we decided to study not only the middle of the fetal period, but the three different stages during the fetal period. 12.5, which is the beginning of fetal period, 15.5, and E18.5, which is just before the delivery in mouse. And also placenta here is a complex tissue with two different major layers, which are called labyrinth here and junctional zone, which has not the same function. Labyrinth is the place where there is the exchange in blood, and junctional zone is more associated to the storage of energy and secretion of hormones. And we learned with Amanda Sferuzzi-Peri to dissect the two parts of the placenta under the binocular loop. It's actually really easy to do. And we did our experiments in the both layers separated. And as you can see here, there is a dynamic of DNA methylation in placenta and also a tissue specificity. Usually, the plain lines here are the labyrinth measurements, while the dotted line is in the junctional zone. And so usually the labyrinths have more DNA methylation than the junctional zone and express more the enzymes DNMT3A, 3B, and 3L. And there is also A dynamics during the placental development with an increase of DNA methylation at the end of gestation, which is linked with an increase of DNMT3A and 3L. And DNMT1 and 3B are downregulated in the labyrinths, particularly during development. And finally, we noticed a diet effect for DNMT3B here in the labyrinths at the beginning of the fetal period, and the same tendency for DNMT3L at the end of gestation. For the enzyme lysine methyltransferase 1A and 1B that methylates the H3K9 lysine, we noticed again a specificity of expression and also a diet effect for KMT1A in the labyrinth at the end of development, and a sex effect here for KMT1B at the end of gestation in the junctional zone. No, in the two tissues, actually. So we noticed in our placenta there is a striking sex differences. They do not react the same to the maternal environment in the same uterus, male and female. Can it be due to hormones or to chromosomes? Well, the hormones is quite obvious an effect. So what about the sex chromosome? Actually, the between sex differences exist even before hormones. Like the cleavage speed is not the same between male and female embryos, and there is differences in pre-implantation development. And in bovine, Bermejo, Alvarez, and colleagues, they show that the gene expression in blastocyst is different if the blastocyst is XX or XY. And they claim that this goes to 1/3 of the genes that are differentially expressed between the two sexes. I'm not sure this is so much, but it means that it's quite obvious that gene expression differences appears really before the hormone secretion. After hormone secretion, there are persistent differences between XX and XY genotypes that you can assess by this animal model, the four-core genotype, where a female, wild-type female are crossed with males that have a deletion of SRY on the Y chromosome, which is called the Y- chromosome, and they have transgene of SRY on an autosome, so they are XY minus SRY. And when you cross these animals together, you can obtain 4 different genotype sex combination in the offspring, with a female that would be XY, so the XY female. There are the XX female that are wild-type animals, XY male that are XY minus SRY, and XX male that are XX SRY. And therefore, you can check the effect of the XX versus XY genotype by comparing in the same sex the two different kind of genotype, and the effect of hormones just by comparing female versus male. And recently they published a paper showing that giving high-fat diet to 14 weeks old mice do not give you the same results depending on the sex chromosome, on the chromosomal sex, sorry. So the weight gain depends on chromosomic sex and not on gonadal sex, which means that actually the XX female gain more weight than XY female, and XX male gain more weight than XY male as an adult. So having 2 X chromosomes is make you gain more weight than having 1 X chromosome. And on the X and Y chromosome, there are the gene pairs that are ancestral homologs. And what's interesting is that these gene pairs here, they have function in gene regulation expressions, such as ubiquitination, chromatin modification, transcription, splicing, and translation. And I will pay attention a little bit more on one of those gene pair that is called JARID1C and JARID1D, or KDM5C and 5D. It's the same genes, they just have 2 different names. They are lysine demethylases of H3K4 trimethylation, And interestingly, Jarin 1C, cadmium 5C, escapes X inactivation mostly in most tissues. And what they did is that they were thinking, okay, if you have 2X versus Y, you gain more weight. Is it possible that this enzyme makes the same? And so they took the knockout model of KMT5C, KDM5C, and they saw that there is the similar phenotype, meaning that when you have one copy of KDM5C, you gain less weight than when you have two copies of KDM5C, meaning that KDM5C is able to make between sex difference in response to the same environment. So in our model, we checked this gene pair, KDM5C and 5D, and we noticed that indeed in placenta, KDM5C is always more expressed than in males, and KDM5D is restricted to male because it's on the Y chromosome. And during development, in both tissue and all along the development, there is the same difference here. So it's not really clear if KDM5C and 5D proteins have the same targets or not. And so, which means, would there be qualitative differences? If there is not, it means that if the expression of KDM5D is able to correct the absence of the less expression of KDM5C in female, it would mean that there would be the same quantity of proteins, same quantity of transcript, and there will be no actual sex differences. So what we did is to design, actually there is one place on the mouse genome that you are able to put a forward and a reverse primer to see the two transcripts in the same time. And it managed to give you good quantitative PCR. And what we saw doing that is that when you study the quantity of both transcripts at the same time, there is still more transcript in female than in male, which means that even if they have the same targets, there is still a difference in expression between the sexes. So the next question would be, is there quantitative differences in H3K4 trimethylation between sexes in the placenta? And that's something we are doing actually. So in conclusion, our results show that placental development is affected by maternal nutrition, and maternal nutrition alters the expression of the epigenetic machinery and the epigenome, with particularly DNA methylation. And there is a dynamic and specific expression of in the different placental tissues of DNA methylation and the epigenetic machinery enzyme expression. And we saw sex differences between male and female. They have different response to the same maternal environment in the same uterus. And we were wondering now, are the sex-specific epigenome leads to different, is it this sex-specific epigenome that leads to the different environmental sensitivity? And what I want to do now, it's not only to describe sexual differences, sex differences, but also to understand the underlying mechanism. And I really think that these enzymes coming from the X and Y chromosome, coming from gene pairs, they can have a really important role in the difference between sex to the same environment. So to conclude, I want to thank all the people of my team. Our team is called MECP2 because in French, MECP2 initials means epigenetic mechanisms of the construction and prediction of phenotypes. And I really want to thank Claudine Junien, which was the head of the lab and is still doing a lot of work in France in the Academy of Medicine to try to make things moving on the sex differentiated medicine. And I want to thank Melanie, which is the engineer of my lab, and she's really a good technician. And I want to thank all the people of the consortium, the Menarini Foundation, for being here today, and I would like to thank you for your attention.